Hi everyone, welcome back to my educational channel on biology. In this video, we're going to discuss a very good trial paper which is SBP 2024 paper 2, and these are the structured questions from question 1 to question 8. Alright, now even if you have tried this paper before, still go through it because I have useful tips to help you to score, and I will comment on how you can use your strategies to obtain maximum marks. Alright, so I'm not just teaching you the answer, giving you the answers, but also teaching you how to score by using specific strategies and answering techniques. All right, so do go through it as a last minute revision. Okay, and then secrets to success involve preparation, hard work, and learning from mistakes. So let's get started. Now, the secrets to high scores and A plus in essay or structured questions. Here are some important guidelines. First, when you look at the question in paper 2, structured question and essay question, do not get anxious, right? Understand the aim and the meaning of the question by reading the question a few times, study the diagrams or statements well, underline the important facts or information that may guide you. Okay, sometimes it's in the diagrams. Circle the important parts of the diagrams and write down some notes beside that. Okay, so that you have some idea of what's going on, and also if you come back and check your work, all those information that you have thought up will still be there. Okay, so write down. Now, I will tell you as we go along, as we look at the diagram. So, biology paper 2 is full of diagrams and statements, and it's an interpretation of the diagrams and the statements that are very important to help you, guide you to answer the question according to the requirement of the question. You must find out what's the requirement, the meaning, the aim of the question. What do they want to test you? Okay, so find out what subtopic or concept, which subtopic or concept is being tested for each question, and always use the keywords from the textbook, important keywords, because keywords are the words that will be marked by the examiner, and this is where you earn your marks. Okay, you earn your marks from the right keywords used. Okay, so you must know the keywords from the textbook, the processes, and then the structures, tissues, and so forth. Now, learn the strategy to successfully answer a type of question. All right, so here, that's why there's one more video coming out. Tomorrow morning, it'll be up, all right, where I will go through a few exam questions, and some of them are new questions, all right, and some are questions that I have also designed myself, all right, based on the experience in composing or creating the whole paper 2, all right, every year for exams. Okay, every year I have to create the paper 2 for school exams and even for district exams. So I'm going to give you some questions that may be asked in the exam, all right? So wait for my video tomorrow on the winning strategy part 3, okay, where I will also teach you some techniques of answering. Now, learn the strategy to successfully answer a type of question and avoid mistakes. All right, there are some mistakes that are usually made, which I will also inform you how to avoid these mistakes, like answering a question too generally, not being specific, and not answering completely. All right, answer halfway, but it's not complete enough to get you the mark. All right, so I'll give you examples as we go along. Now, so some of the strategies are as follows. Comparison mode. You can use the comparison mode where you need to write, the rate of A will be more or maybe less than the normal rate, and so forth. Something may increase, level of something may increase or decrease. All right, that means you're comparing it with the original state or the normal state. Okay, and sometimes you have to use the not mode. When something is not present, an organelle is not present in the cell, what will happen? So you must write. Then there will be the cell cannot undergo respiration or photosynthesis and so forth. Okay, the not mode. Now, or another strategy is event, process, result, or effect mode. Now, this one I will teach you tomorrow, okay, so that you can answer smartly and know exactly what the question is requiring of you. Now, determine how marks are allocated for a question, all right, so that you can make sure that you have enough facts to get you all the marks. For example, if you're asked to compare two processes, you must state at least one difference and at least one similarity, okay, and make sure you look at the total marks to give enough similarities and differences, right? Now, always write more points than what is needed, especially for the essay question. A structured question also, you can write 1 or 2 points more, okay? But don't fill up the whole page with answers. just that you can write a few lines down, and for essay question, there's no limit. Okay, so write more points so that you will ensure that you get the maximum score in case some of your points are not correct. Okay, right. So with that said, let's go on further. Now, analysis of chapter 2, cell biology and organization, because we're going to discuss one question on that. So topics that have come up are on mitochondria, Golgi apparatus, damaged plasma membrane, that is in SPM 2022, ribosome, Golgi apparatus, extracellular enzyme, blocked bile duct, function of mitochondria in 2024. So if you want to look and see how these questions are asked and whether you can answer them, please do try the questions in the past year question book. I hope that you have done it already, because it's now it's a few days before the exam, right? So if not, you just try these questions here. Let's look at the first question. The topic is from chapter 2, cell structure and organization. Now, diagram 1.1 shows component P in an animal cell. All right, this is component P, and there's structure Q also. State the name of component P and structure Q. Okay, so component P, you can see, is this organelle here, round organelle near the center of the cell. So what is component P? The nucleus, right? And structure Q are the long, thin, thread-like structures. They're actually the chromatin, or if you write chromosome, is correct also. So chromosome and chromatin are the same thing, just that chromatin are long and thread-like chromosomes, all right, before they condense and before they are visible in the stage of mitosis. Now, number 2, state the function of component P. Okay, so function of organelles. You must know all the functions of the organelles. So this is worth, I think it's only 1 mark, all right? So any one function. Now, firstly, P controls all the cellular activities. This is the main function. And if it's 2 marks, you must write another function if it's 2 marks. P contains deoxyribonucleic acid, or DNA. Now spell this out in full. Or if you don't want to write deoxyribonucleic acid, you can write genetic information, which determines the cell's characteristics. Let's go on to B. Diagram 1.2 shows the components of nucleic acid found in structure Q. Okay, found in structure Q. This structure here, the chromosomes, there's this nucleic acid, right? So you can see 2 strands of polynucleotides on the left and right, and then we see the letters ATGC, which stands for the 4 nitrogenous bases: adenine, thymine, guanine, and cytosine. Question: state the type of nucleic acid shown in diagram 1.2. Type of nucleic acid. How many types of nucleic acids are there? There are 2 types, right? The RNA ribonucleic acid and the DNA, deoxyribonucleic acid. So for RNA, there's only 1 strand of polynucleotide, but here you have 2 strands. So this is DNA. Now spell out in full. Okay, to be safe, you better spell out in full. Deoxyribonucleic acid, and only in brackets put DNA. All right, don't just leave it as DNA. You may not get your mark. So spell out in full. Now, next question. Nitrogenous base of the nucleic acid structure in diagram 1.2 forms the genetic code that is important as a carrier of hereditary information. The nitrogenous bases A, T, G, and C. Okay, they form the genetic code in the form of triplets. All right, 3 bases form the genetic code for 1 amino acid that needs to be connected to form. It needs to be joined to form the protein or polypeptide chain. Explain the effect on the characteristics of organism if there are changes in the sequences of the nitrogenous bases. Okay, so here I want to teach you the strategy of answering. Firstly, you have to identify the event. The event is that triggers everything. The change that triggers the process is changes at the sequences of the nitrogenous bases. For example, maybe a sequence of AGC here, it may change so that maybe G is replaced by a T or another base, right? So there's a change in the sequence of the nitrogenous bases, right? That's the event. Then what are you supposed to answer? You have to explain the effect on the characteristics of an organism, all right? If there are these changes at the sequence of the nitrogenous base. So underline this part. characteristics of organism. You have to mention something about the effect on the characteristics of the organism. So your answer must start with that. All right. So the characteristics of the organism or an organism will be changed if there are changes at the sequences of the nitrogenous bases. All right. But you don't have to repeat the event. Then after that, you go backwards and explain how you arrive at this effect. Okay. What happens after the event occurs? What did it trigger? What process did it trigger? So that's the step-by-step process. That's the process that you must explain. So that means event causes the process, the step-by-step processes leading to the final effect, right? So you write the effect first based on the question, and after that you move backwards and explain this is because what happens after the event. So after the event occurs, changes at the sequences of nitrogenous bases, we say that mutation has occurred, which are spontaneous and also abnormal random changes that occur to the genetic material. Okay. Now, when mutation occurs, the genetic code used to synthesize the protein is changed. For example, the triplet code AGC may be changed to ATC. Once the code is changed, that means the amino acid that is used to build the polypeptide chain will be changed. Okay. So this leads to changes in the amino acid sequence used to build the protein structure. So the protein structure will also be changed. Okay, either you say change in amino acid sequence or change in protein structure. Thus, abnormal, non-functional proteins are produced, and if you write further, you can say that it causes genetic disease. Okay, now, so from P2 to P5, this is the process that occurs that leads to P1. All right, so write the effect first and then followed by the process. But before that, you have to identify the event. So if it's 2 marks, then any 2 points, but P1 must be in because they already ask you what happens to the characteristics of the organism. So P1 is a compulsory answer. Okay, and then any one of these answers. So here's the strategy. How are you going to break down and analyze a question in order to understand it fully? In case, sometimes in the question, there are lots of words and statements made, and you get a student may get confused. What is the question trying to ask me, right? So you read the question when it comes to state the effect or explain the effect. So you find first of all the event, okay, and what effect you are going to speak on. All right. So the event is the happening that triggers the whole process leading to the final effect. So there are 3 parts. Event, then process, leading to effect. But first, you identify the event in your question. Next, they will ask you, when you answer, they'll ask you, state the effect of the event on something. Just like just now, state the effect of the changes to the base sequence in DNA on the characteristic of an organism. So you have mentioned the characteristics of the organism will change, right? That is what we mean by state the effect on something first. And after that, you explain the process. Then you say this is because mutation has occurred. That means you are explaining the process after the event. Okay? This is because after the change in the nucleotide base sequence, then mutation has occurred, and this will change the genetic code and the type of amino acid. Okay? That's used to build the polypeptide chain. Alright? So step-by-step process must be explained to gain the maximum number of marks. So here's how you can do it in your question paper. To identify the event, just underline or bracket the part and put that event or EV, and then identify the effect. What effect are you going to talk about? So effect on the characteristics of the organism. Right now, that's the effect. So the event is if there's a change in the base sequence in the DNA molecule, like what was mentioned just now in the first question, that's the event. Then what is the effect on the characteristics of the organism? All right, so label there EF or effect. And then after that, you answer the question by stating the effect first, right? That the characteristics of the organism will be changed. Then only you talk about the process, okay, that occurs once the event occurs. What's the process that occurs at P2 to P5, right? Until it leads to the effect, which is the characteristics of the organism will be changed, okay? Now, next question is from Form 5 Chapter 2 on Organization of Plant Tissues and Growth. So in the past years, questions been asked on shoot tip zones, like the zone of cell division, zone of cell elongation, and zone of cell differentiation. And then primary and secondary growth, and why we trim the shoots, right? Also, root hair cell drawing has been asked. So let's look at the question. Now, diagram 2.1 shows a component in a plant cell. Name the structure X and Y. So what is this plant cell? Yes, it's the chloroplast, right? Chloroplast. You can see the disc-shaped structures inside the chloroplast and the double membranes, outer and inner membrane. So what is the name of the structure? Name of structure X and Y. Now, X is the fluid, okay, in the chloroplast. So X is the stroma, okay, and then Y is the disc-shaped structure, but it's only labeling one, okay, so it's called the thylakoid. If the whole set of thylakoids is considered, then it's called a granum, okay, granum, G-R-A-N-U-M. Now, state the importance of structure X. So, what's the importance of the stroma? Stroma is the location where something happens right now. So it is the site of the light-independent reaction of photosynthesis, where there is reduction of carbon dioxide and glucose production. All right. So any one of these site of light-independent reaction of photosynthesis, or site of reduction of carbon dioxide, or site of glucose production. Okay. So if you are not sure, write down a few, right, to get that mark. Now, explain the effect of optimum surrounding temperature on the rate of photosynthesis in plants. Okay, so let's say this is 2 to 3 marks. Okay, I didn't write this down. It depends on the marks. If it's 2 marks, then you can afford to write less, but if it's 3 marks, then more. Okay, now again, the effect of something on something. This is again the strategy. Okay, so break it down into event, process, and final result or final effect. So the final effect must be on the rate of photosynthesis. The event is the optimum surrounding temperature. Get it? Event is optimum surrounding temperature. Rate of photosynthesis is your final result. What happens to it is your result. Okay? So first of all, state what happens to the rate of photosynthesis if there's optimum surrounding temperature. So the rate of photosynthesis will increase to the maximum level, right? Now, here we use the comparison mode, increase, or will be at the maximum. Okay, the rate of photosynthesis will be at the maximum because it's the most suitable. Optimum surrounding temperatures means the most suitable temperature. Somewhere around 30 degrees Celsius, somewhere around there. So because now then you have to state the reason why again, right? Why is it that the rate of photosynthesis increases when the surrounding temperature is optimum? Because the activity of the photosynthetic enzymes, this is the important thing they want. Okay, photosynthesis is controlled by, is carried out using photosynthetic enzymes. So when it's at the optimum surrounding temperature, the photosynthetic enzymes are reacting at the optimum rate or maximum rate. Okay, so the activity of the photosynthetic enzymes increases, and photolysis of water or reduction of carbon dioxide also increases. Okay, so especially the reduction of carbon dioxide, it uses enzymes, all right, and also for photolysis of water. So these two stages of photosynthesis will increase when the surrounding temperature increases, because photosynthetic enzymes are involved, and photosynthetic enzymes are affected by the surrounding temperature. Okay, now number 4, the fourth question. Diagram 2.2 shows the changes in the leaves color of maple tree during 2 different seasons. State why there's a change in the color of the leaf during autumn. Okay, during autumn, underline that. So again, we look at from the aspect of event, process, and effect or final result. Now the event is during autumn, and the final process or effect, the final effect is change in the color of the leaf, right? So you have to connect the two together. During autumn, what happens so that there's a change in the color of the leaf from green to yellow, right? What happens? So it's something to do with the temperature and the light intensity, right? The change in temperature and light intensity when the day or when the season changes from summer to autumn. So during autumn, light intensity and temperature becomes lower than in summer. Okay, then the plants stop making chlorophyll, or chlorophyll is broken down into smaller molecules because of the lower temperatures and light intensity. That's the signal for the plants to stop making chlorophyll, because they are going to go into a dormant state, really, into a resting stage when winter comes. Okay, they will not be actively carrying out photosynthesis in winter because the temperature is too low. So they stop making chlorophyll, and the chlorophyll is broken down into smaller molecules. So then the chlorophyll level will drop. So without chlorophyll, the yellow pigments or carotenoids, which can be seen on the leaf here, which is always produced, will become visible in the leaf. It's the chlorophyll that is broken down and that the plant stops making, but the carotenoids are still there. Okay, so without chlorophyll, the yellow pigments called the carotenoids, which are always produced, will become visible in the leaf. Okay, so this is a question that hasn't been asked before. I can come up the SBM. Now, the third question is on Form 5, Chapter 8 on biodiversity, and the question that has been asked in SPM is in the year 2022 when they test the student in question 11. Okay, it's a compulsory question on the meaning of animalia, dichotomous, key, vector, and pathogen. Okay, so let's look at this new question. All right. Now, diagram 3 shows two types of microorganisms, X and Y. So based on the diagram 3.1, state the types of microorganisms X and Y. All right, based on diagram 3.1, state what is X and Y. So X can see got flagellum and it's a single cell. All right, it's a microorganism and it can move. So what is this microorganism? It's bacteria. Okay, and this one you can see the budding. Right, so this is actually like yeast, but we are asked to state the type of microorganism. So you cannot write down yeast. There are 5 types of microorganisms right now. So it is bacteria, algae, fungi, protozoa, and virus. Right? So this one is fungi. So if you write yeast, it's wrong because they want the type. So read the question carefully. Now, next, microorganism X is Vibrio cholerae. This one. State the disease and symptoms caused by this microorganism. So this bacteria is very dangerous, and from the name we already know the disease. The disease is based on the last name. What is it called? Cholera, correct? So this disease, what's the symptom? This will cause a person to have diarrhea, chirik-birik, or latuj, very serious diarrhea. So diarrhea, the spelling is D-A-I-A-R-R-H-O-E-A, okay? Or you just write down vomiting and dehydration. Okay? This is from Form 5, Chapter 8 on biodiversity. Now next, microorganism Y is involved in bread making process. This one. State the role of Y in the process. How does Y help in the bread making process? Okay, so this is related to another chapter, which is Chapter 7 on respiration. Okay, it's only 1 mark. So it could be 1 or 2 marks. So Y is yeast, which carries out alcohol fermentation. Chapter 7, Form 4, Chapter 7. Glucose is broken down incompletely without the presence of oxygen to produce carbon dioxide, ethanol, and energy, and then the trapped carbon dioxide causes the dough to rise and makes the dough soft and fluffy. Okay, so you must name the process first and then explain what happens in the process. So we have the acronym for this equation. Do you know what it is? Yes, it's GEL. Sorry, it's GC. GC, not GEL. GEL is for lactic acid fermentation. So GC is for alcohol fermentation. G arrow CEE. G arrow C is for carbon dioxide, E is for ethanol, and the other E is for energy. GC. That is for alcohol fermentation in yeast. So you can remember the acronym and you'll be able to recall the process so that you can write down all these substrates and the products easily. Okay, GC for alcohol fermentation. So this topic on Chapter 7 is likely to come out this year in 2025 because it has not been tested before. It's the only chapter that has not been tested for the 5 years, until now, okay, for the 4 years. Now let's look at this HOTS question, all right? An individual has been infected with acanthamoeba, keratitis disease after wearing contact lenses that have been cleaned by using tap water. This disease is caused by a protozoa, and ophthalmologists suggest this individual to use the solution as shown in this diagram. It says here cleanser, all right, especially for washing the contact lens, right, as shown in the diagram. Explain, that means explain why the ophthalmologist suggests this special cleanser, right? And what happened to the person? How did he get infected by the acanthamoeba keratitis disease? All right. So disease caused by protozoa, and the contact lens was cleansed with tap water. So probably the protozoa is in the tap water, right? So infected the lens, and after that infected the eye of the person who wore the contact lenses. That's how it's transmitted. Okay. So you're asked to explain why the ophthalmologist suggests using this cleanser. So first you explain what happened to the person. Tap water is the habitat of the protozoa. So the protozoa is an aquatic microorganism that has infected the lens and the eye. Okay, aquatic microorganism. So the solution used is a sterile solution free from microorganisms. That's the meaning of sterile here, free from microorganisms. So it's very clean, and then sterile is the important word here. Okay, the solution probably contains some disinfectant that can effectively kill the protozoa when the lenses are soaked in it. All right, that is to get rid of the protozoa. And yeah, it's very important for those who wear contact lenses to take care of the cleanliness of the lens and don't simply wash the lens with tap water. However, all this information here is not intended as medical advice. So there are solutions that 3% hydrogen peroxide acts as a strong disinfectant that really kills the amoeba, but it is toxic to the cornea. Okay, it's found to be toxic to the cornea. So don't simply use any kind of cleanser that's bought off the shelf. You must always consult an eye specialist before using all this if you are wearing contact lenses. All right. Personally, I've used contact lenses before. I also got infected by some kind of something like a cyst also in the eye. So I was advised not to use any more contact lenses. And after that time, the problem disappeared. Okay, so nowadays I just wear specs. Now next, Form 4, Chapter 7, Cellular Respiration. Okay, so no SPM questions have been asked yet on this topic. Get ready for it this year. Okay, so I've done the analysis myself for all the SPM questions. Okay, it was a lot of work, and no questions have been asked on Chapter 7, Cellular Respiration. So this year, you can be asked on, say, aerobic respiration or fermentation, such as alcohol fermentation or lactic acid fermentation. You can be asked on the products of these types of fermentation, and then comparison between these 3 different types of processes. Okay, so get ready for it. In case it comes out in question 11, it may come out, because question 11, not necessarily only certain topics will come out from there. All right, as I will explain later on. Maybe tomorrow I will explain something about question 11. Okay, now revise the topic thoroughly, this topic, and also, of course, you also revise other topics, not just concentrate on one topic. That is setting yourself up for failure if you just revise one topic. Okay, revise all the topics to make sure you have a wide scope of questions that you can answer. Revise all topics. Now, number 4, diagram 4.1 shows respiration process that occurs in a cell. So this is topic is on Form 4 Chapter 7, as we said just now. Now, glucose through process K becomes pyruvate, and then it produces, with the help of gas X, then the process occurs which releases carbon dioxide and water, and the energy is 2898 kilojoules per mole of the glucose. Okay, so identify process K and gas X. Glucose is broken down into pyruvate. What's this process called? Yes, it's called glycolysis, the breakdown of glucose. Okay, the glyco is for glucose, lysis means to break down. The breakdown of glucose into pyruvate. Now, what is gas X that helps to further break down the pyruvate? Is oxygen, correct? So this is actually the process of aerobic respiration. All right, diagram 4.1. Now next, state the advantage of the respiration process shown in diagram 4.1 in a human muscle cell. Advantage, see, you can be asked on advantage. So the advantage is very clear, because a lot of energy is released, right? So more energy, which is 2898 kilojoules of energy per mole of glucose, or 38 ATP is produced. You can use this number here, all right? Now, the breakdown or oxidation of glucose occurs completely in the presence of oxygen, okay? And then lactic acid that causes muscle cramps is not produced. So that's the advantage, all right? And more energy is released through the complete breakdown of glucose. That is aerobic respiration. So that's the advantage. Next. Okay, this is a hot question. It looks difficult, but actually it's very easy if you read the question carefully and understand what the question is about. Okay, so a simple respiration experiment was done by placing blended bananas with yeast in a closed container. The alcohol content is measured every day and the data is plotted in a graph as in diagram 4.2. Okay, so it's a new graph for you, right? So read the graph carefully. It's the volume of alcohol against the time. Okay, now next, state the difference in the process that occurs before day 4 and after day 4 of the experiment. Now, before day 4, let's read. The level of alcohol produced is 0, okay, until day 4. After that, the level of alcohol starts to increase. That means alcohol starts to be produced after day 4. So why is it that there's no alcohol produced before day 4 and after day 4 there's alcohol? Surely the process is different, right? So before day 4 now, if you read back, the banana must contain some substrate like glucose or some other sugars, right? With yeast in a closed container. Underline the word closed container. So the alcohol content is measured. Now, in the closed container at first, is there oxygen? Yes, there's oxygen, right? But after a while, the oxygen will be used up, okay? So before the oxygen is used up, it's probably from day 1 to 4. What is the process that occurs? It's aerobic respiration using oxygen because there's enough oxygen. After that, when the oxygen supply is already used up, then the yeast has no choice but to carry out fermentation without oxygen present, and that is alcohol fermentation. So here they're asking you the difference between process that occur before day 4, that is aerobic respiration, with the process that occur after day 4, which is alcohol fermentation. What's the difference? 1 mark only, right? Difference between, so choose one difference, one main difference between aerobic respiration and alcohol fermentation. Now, since it's 1 mark, let's see how to answer first. Before day 4, yeast undergoes aerobic respiration, but after day 4, yeast undergoes alcohol fermentation. Now, that's the difference, okay? Or before day 4, complete oxidation and breakdown of glucose occurs. After day 4, incomplete oxidation or breakdown of glucose occurs. Before day 4, there's sufficient oxygen. After day 4, there's insufficient oxygen. So actually, it should be the first point, and then the second point should be P3. And then if you write it in sequence, and third point is P2. All right? When there's sufficient oxygen, then there's complete breakdown of glucose. When there's insufficient oxygen after day 4, then there's incomplete breakdown of glucose. If you're not sure, write at least 2 points. Okay? Now, another hot question, which looks difficult, but actually it's very simple. Experiment is repeated using an open, highlight this word, open container. Now, just now was closed container. So you must be able to pick up the words that are important from the explanation. So explain the effect on the volume of alcohol in the container. Again, we have to break it up into 3 parts. What is the event? What is the event? The event is the repeating the experiment using open container. Open container means what is present? Oxygen is always present from the air. You can go into the container that's open, right? And that is your event. And then what is the final result? You have to tell what happens to the volume of alcohol in the container. The effect is on the volume of alcohol in the container. So mention this first, okay? Now, what happens to the volume of alcohol in the container? Will it increase? Will it decrease? Yes, the volume of alcohol will be 0. The volume of alcohol that is produced will be 0. No, that is produced will be 0. Okay? So, no alcohol is produced. And then you're asked to explain. So that means after you explain this, why you have to explain why, the reason why this no alcohol is produced when the container is open, right? So when container is open, there is oxygen supply from the air that's sufficient, right? P4 first. Okay, sequence. So oxygen supply from air is sufficient, or from the atmosphere is sufficient. So yeast carries out aerobic respiration in open container. And there's no alcohol fermentation. Alcohol fermentation does not occur because oxygen is present. Alcohol fermentation only occurs when oxygen is absent, right? So because there's aerobic respiration, the breakdown of glucose occurs completely and no alcohol is produced. Okay, no alcohol produced. You can write here no alcohol produced. It's already in the first point. Okay, so if you're not sure, you can write down a few of these points. Next, another question from Form 4, Chapter 5 on metabolism and enzymes. So in the past, the lock and key hypothesis, anabolism, catabolism, effect of mercury as inhibitor has been tested. Now, let's look at this question from 4, Chapter 5. Diagram 5.1 shows the stages in the sucrase enzyme reaction. Okay, sucrase. So here you see the sucrase. So look at the diagram first. Understand what is showing. Sucrase. So this is an enzyme, right? The word ASE. Now the substrate is this one. Okay, substrate P, and then they don't show complex Q. You probably have to draw this, right? And then the end, they show the final products. The substrate P has broken down into 2 parts. Okay. Now state the name of substrate P. If the enzyme is sucrase, what is substrate P? Yes, it must be sucrose. Okay. So the substrate P is a disaccharide called sucrose. Okay. Now draw the complex Q in the space provided in diagram 5.1. Asked to draw. It's very easy, right? You just combine these two together to form the complex enzyme substrate complex. Okay, like this. Very simple. So use a pencil to draw the shapes. Just make sure that the 2 parts are combined at the active site here. This is the active site. This one and this one. Now, next question. Diagram 5.2 shows a graph of the enzyme reaction rate against temperature. So rate of enzyme reaction against the temperature, right? Now, question. Explain the differences between the rate of enzyme reaction at point M and point N. Right? Point M and point N. Rate of enzyme reaction. Differences. Okay? So we can see here that at point M, the rate of enzyme reaction is the highest. At N is the lowest. And you're asked to explain. 3 marks. 3 marks for this. Okay? So first of all, you state that the rate of enzyme reaction at point M is much higher than point N. Alright? Then only you explain why. So at point M, the temperature is 40 degrees Celsius. The enzyme is most active because this is the optimum temperature, okay, for the rate of reaction. Alright? Somewhere around 37 to 40 degrees Celsius. And then at point N, why is the rate lower? Because the enzyme is denatured at high temperatures of 70 degrees Celsius. Alright? Denatured. So the rate of reaction will drop to 0. Now, the frequency of effective collisions at M is more than N. Or at M, the frequency of effective collision is maximum, whereas at N is minimum. Okay? So 3 marks. Now, hot question again. Diagram 5.3 shows the extraction of agar from seaweed. So the seaweed is put into the container where there are beads with the enzyme. So this is the enzyme technology called immobilized enzyme technology. Okay, the enzyme is attached to the beads. Okay, then out comes the agar-agar. This is the final product. Okay, let's read the question. Enzyme immobilization technology is used in various industrial applications, such as the extraction of agar from seaweed. By using a suitable example of immobilized enzyme based on this, explain how the agar is extracted. So what is the enzyme involved in breaking down the cell walls in the seaweed to release the agar? What's the enzyme? To break down the cell wall. So it must be cellulase, right? Cellulase to break down the cellulose cell wall. Okay, so the first answer, because you have to state a suitable example, okay, of the immobilized enzyme. So the enzyme used is cellulase. This is 1 mark. Now for another 2 marks, let's see how to write. You are asked to explain how the agar is extracted. So all this is taken from the textbook. All right, it's in the textbook. So the enzyme combines with the inert and insoluble substances in the beads, to increase the resistance of the enzyme towards changes in pH and temperature, so that it can be stable and it will not break down or be destroyed, okay, due to changes, or it will not be denatured due to changes in pH or temperature. So the enzyme molecules remain the same position after the reaction, all right, and they can be easily separated from the product. They will not come out together with the product, okay, they will remain in the beads there, all right. So the enzyme can break down more cellulose in the cell walls of seaweed into glucose to release more agar from the cells. So because they remain in the same position, so the seaweed can be more seaweed can be inserted for the process of breakdown by cellulose, all right. And then when the actually what happens after the cellulase breaks down the cellulose cell walls, the agar will be released from the cells, okay. So cellulase is used to break down the cellulose cell walls of the seaweed to release the agar. Now here's another hot question which may be asked, because this technology has not been tested in SPM, the immobilized enzyme technology. So get ready to be asked about it. What are the benefits of immobilized enzyme technology? 3 marks. This is actually partly in the textbook. Okay, so one benefit compared to the old enzyme usage, where there is no beads, no beads to attach the enzyme to. Okay, so the benefits of this new technology is that it allows repeated use of the enzyme, all right, and the enzymes are always remaining in the same position, so they can be used again and again. Improves the stability of the enzyme, increases resistance of enzyme to changes in pH and temperature, okay, straight from the textbook. And so that means the enzyme will not be denatured by changes in pH or temperature, increases agar yield or yield of product, okay, reduces contamination of the enzyme or of the product, right, and contamination of product by enzyme, and it's an eco-friendly method that will not harm or destroy the environment. So I would choose, let's say you ask to give it's 3 marks, so three benefits. I'll choose the first one, second, third, and fourth. Okay, not sure. Okay, until the fourth one, or you can put until the fifth one. These are the basic answers. Right. So the next question is from 4 chapter 9, nutrition and human digestive system. What has been asked is on physical and chemical digestion, the mouth, gastric bypass, obesity, anorexia, bulimia, muscle dysmorphia. Okay. Now, diagram 6 shows organs involved in human digestive system. State the enzyme found in R. Okay, R is the stomach, right? So what's the enzyme? Pepsin, right? That breaks down the proteins. All right. Now, all the enzymes of proteins end with sin. Sinful. It's full of sin. Okay, the sin, sin. All right. All the enzymes of proteins have the word sin. Okay, pepsin, trypsin, and erepsin. Okay, and protein has the in there. So okay. So R is pepsin, and the inactive form is pepsinogen. All right. Now next, state the food class that is hydrolyzed by the enzyme in A1. What food class is it? So it's proteins, right? Food class, that means you must mention like whether it's protein or carbohydrates or lipids. So it's protein. Now the following statement explains about the health condition experienced by Dr. M. By Mr. M, not Dr. M. Mr. M has experienced abdominal pain in the lower right side. The doctor found that his appendix had ruptured and caused Q to be infected. Q is this part here, right? As a result, a part of Q had to be cut and removed. So what is Q? Now, Q is the lower part of the small intestine, all right? Not the appendix, it's the lower part of the small intestine. The appendix has ruptured here, so the Q infected and the whole thing is cut up, all right? This has happened to people before, right? When the appendix ruptured or burst. Now, explain the effect on nutrient absorption. That is, if P had been cut and removed. So again, we have to break down into the event, process, and the effect. Now, the event is that part of Q had to be cut and removed, was cut and removed. So now the ileum, which is the last part of the small intestine here, is shortened. Okay, P is duodenum, and then you have duodenum, and then the last part here is ileum. So the ileum has been shortened. Okay, that is the event. Now, what is the effect on nutrient absorption? Okay, so straight away when you think, you know already. Logically thinking, if the ileum is shorter, then the nutrient absorption, which is carried out by ileum, will be reduced, right? So less nutrients will be absorbed, or nutrient absorption. Follow from here. Nutrient absorption decreases. You have to compare with the original situation. So use the comparison mode. Compare the original situation. And you can use the words like less, more, or decrease. Don't just write down nutrient absorption. Nutrient absorption will carry on. You have to write whether it decreases or increases. So decreases, and then after that you give the explanation. Okay, that means you have mentioned the event and the final result. Now the process in between. What makes the nutrient absorption decrease? The reason is the ileum becomes shorter. Number of villi decreases when you cut the ileum, right? Cut the ileum or Q out, part of it out, then the ileum becomes shorter and there's less villi or less of the finger-like projections. So the total surface area for nutrient absorption will decrease, right? And the number of blood capillaries and lacteals for absorption and transport of nutrients also decreases. So any three of these. The first three is the best. Okay? Talk about the total surface area for nutrient absorption decreasing because the ileum will be shorter. Okay? So differentiate digestion that takes place in P and R. Now P is the duodenum after the stomach, and R is the stomach. Okay? So 3 marks. Now, for digestion in different locations, this is how we can state the differences. We compare based on these aspects. Tip for digestion, compare from aspects of the digestive juice, the digestive gland involved, the enzymes involved, then the whole equation. All right, what are the substrates? What are the enzymes? What are the products? And also the pH involved at that location. Okay, so let's look at the answer. Okay, now, since there is no table given for this structured question, don't go and draw a table. You cannot simply draw a table in the structured question section if there is no table. But you can draw a table for essay. All right, but if there's no table in the structured question, don't go and make a table out of these lines. So in this case, you have to compare using the words but or whereas in a statement. Just list down the statements, the differences. Okay, so the first point is pancreas secrete pancreatic juice into P, which is the duodenum. But in the stomach, it's the gastric glands that secrete gastric juice into R, which is the stomach. Okay, notice that I mentioned P and R. I didn't just mention duodenum and stomach because the question talks about P and R, so you must mention P and R. Although you can state what it is, what are the organs. Now, second point is from the aspect of we have compared the digestive juice, so now we compare the enzymes. P contains the enzymes lipase, pancreatic amylase, and trypsin. Think of the word LAT, L-A-T. Lipase, and then A for amylase and T for trypsin. But R contains the enzyme pepsin. Okay, the main enzyme, one of the main enzymes, pepsin. So third point is on the substrates. Okay, in P, the digestion of starch, lipids, polypeptides occur. Right, but in R, digestion of proteins occur. All right, now next talk about the products of digestion. Okay, you've mentioned the substrate, enzyme, now products of digestion. So in P, the products of digestion are maltose. Okay, when pancreatic amylase is broken down, it produces maltose. Fatty acids. Okay, when the lipase breaks down the lipids and glycerol, and then when trypsin breaks down the polypeptides, you form the peptides. Okay, so these are the products of digestion in the pancreas. Sorry, in the duodenum. In P is the duodenum. But in R, the products of digestion are polypeptides. Okay, because the pepsin breaks down the proteins into polypeptides in the stomach. That is R. Okay. Now in P, which is your duodenum, enzymes function in an alkaline medium of pH 8. but in R, enzymes function in acidic medium of about pH 2 because of the hydrochloric acid. All right. So this is a good question, good hot question, may be asked in the SPM. All right, take note of this question. Now, next question is from Form 4, Chapter 15, Sexual Reproduction, Development and Growth. So previously in the SPM, topics that have come out have been talking about the comparison between, have been asked about the fetal vein blood and maternal artery blood, okay, seminal visceral. But the question is a hot question, so it's not directly asked, okay. IVF, artificial insemination, okay, it's a hot question. So you have to try the question to see how they ask it. It's asked in an indirect way, that's why it's more difficult, right? So we'll look at some of these type of questions that are asked indirectly. Now, these type of questions can be asked because reproduction topic so far, spermatogenesis and oogenesis has not been asked, okay. Diagram 7.1 shows the process of human spermatogenesis. So this is the spermatogonium, then it undergoes meiosis, right? So what is this cell here? What is this cell that undergoes meiosis? It's the primary spermatocyte. Then after meiosis 1, it becomes what? Secondary spermatocyte. It's not named, so you can write it down when you are looking at the diagram, okay? But you must remember your facts. And after meiosis 2, spermatids are produced. Then the spermatids will undergo differentiation to form the sperm, okay? So state the name of cell P here. Now, the cell before this is not labeled. This is actually primary spermatocyte. And this will be the secondary, primary from primary to secondary. The primary spermatocyte undergoes meiosis 1 to produce the secondary spermatocyte, okay? And then next, state the number of chromosomes in cell P. So the number of chromosomes in cell P will be half that of the cell above it, the before meiosis. Because meiosis will reduce the number of chromosomes, all right, to the haploid condition. And this is for humans. So humans at first will have 46 chromosomes in the diploid parental cell. And then after meiosis 1, it become half, okay, so 23 chromosomes, all right, it's a haploid cell. The secondary spermatocyte is a haploid cell after meiosis 1. Now, explain the effect on the quality of sperm produced if process Q does not occur. This is a hot question. So again, note your event and then the process and then the final result. Okay, you see the event and the final result is mentioned. Okay, now the event is process Q does not occur. All right, event is process Q does not occur. Then the effect is on quality of sperm. So talk about the quality of sperm first. Will the quality of sperm can either become better or worse, right? Okay. So quality of sperms will decrease. Okay. Why? Because process Q. What is process Q? Go back to this process. What is process Q? Differentiation, right? So mention it first. Process Q is differentiation. It's good to identify the process first. All right. Differentiation means the process whereby the spermatid, which is circular in shape, will change its shape and structure to form the sperm with a head, midpiece, and tail, so that it can carry out its function well to fertilize the ovum, to swim towards the ovum for fertilization, right? So that's differentiation, the change in the structure and shape, so that it can carry out its functions well. So if Q does not occur, the quality of sperms will decrease. Now that is to answer the first part of the question here. And then we go back and talk about what happens when process Q does not occur until it causes the quality of sperms to decrease. So after process Q does not occur, what happens? The spermatids will not be able to differentiate into the sperms with head, midpiece, and tail, right? So there'll be less mitochondria compared to normal sperms also, because it's part of differentiation. So without the tails, without mitochondria, the abnormal sperms are not able to swim towards the ovum in the fallopian tube for fertilization. That's how the quality of sperms will decrease, so that maybe the sperms cannot fertilize the egg, right? Okay, so you can see here that it's step by step. First, you mentioned spermatids do not differentiate, then less mitochondria, no tails also will cause the abnormal sperm not to be able to swim to the ovum. That's how the quality is reduced. Quality of sperms is reduced. So Take note that I use the comparison mode and the not mode. The comparison mode is when I use decrease, reduce, and so forth. Not mode, you see, spermatids do not differentiate. Sperms not able to swim, less mitochondria. Okay, so all this must be used, because if you don't use this, it doesn't make sense. You need to compare with the quality of the sperms before, in the normal situation where Q occurs. All right. Now next, diagram 7.2 shows the behavior of chromosomes during meiosis. State two differences between behavior of chromosomes in diagram 7.2 and the behavior of chromosomes at anaphase. Now look at diagram 7.2. It shows you homologous chromosomes separating. You can see that each chromosome has got the two arms and the two sort of leg-like structures. It's the X-shaped chromosomes that separate. It's not the sister chromatids. Take note, it's not sister chromatids that separate. It's the whole homologous chromosomes that separate from each other, right? And move to opposite poles. So actually there should be two, the size of the chromosomes should be different, not all the same sizes. Okay, so there should be 4 sizes of chromosomes. Now, state two differences between the behavior of chromosomes in diagram 7.2 and the behavior of chromosomes at anaphase. Anaphase means it's for mitosis, because there's no anaphase 1 or 2, so this is anaphase of mitosis. Okay, so now the table is already there for you, so you just fill it up. Two differences between behavior of chromosomes. Just talk about the chromosomes. Okay, so for this phase, what phase of meiosis is it? The phase is called anaphase what? Anaphase 1, because homologous chromosomes separate. So it's anaphase 1. But you're not asked to write down, identify the stages. But you must know. Diagram 7.2, you can write in pencil first, you are not sure. Anaphase 1, in case you forget. And then anaphase of mitosis. Okay? Anaphase 1 meiosis, anaphase of mitosis. Okay, what are the differences between the behavior of chromosomes in diagram 7.2 here, in anaphase 1, and then in anaphase? So first difference is that in anaphase 1 is the homologous chromosomes that separate from each other. But in anaphase of mitosis is sister chromatids that separate from each other. Correct? And then the second point is you must go step by step. After they separate, where are they put? Put to opposite poles. So in the P2, the homologous chromosomes are put to opposite poles, and as a result will form 2 haploid daughter cells at the end of the meiosis 1. But in anaphase of mitosis, it's the sister chromatids that separate, so they are put to opposite poles, and as a result, 2 diploid daughter cells will be formed at the end of mitosis. Alright, because each chromatid will become a whole chromosome. Alright, diploid cells are formed at the end of mitosis. Next, non-disjunction conditions that occur during meiosis can result in abnormal number of chromosomes in gamete. Explain how this happens. Non-disjunction, as we know, means that homologous chromosomes fail to separate in anaphase 1, or sister chromatids fail to separate in anaphase 2. Okay? It can result in an abnormal number of chromosomes in the gamete. Explain how it happens. So you have to explain the non-disjunction. Alright? Now, in non-disjunction, homologous chromosomes fail to separate in anaphase 1, or sister chromatids fail to separate in anaphase 2. So how does it result in abnormal number of chromosomes? So this is because spindle fibers fail to form during meiosis. This just explains the first part. Thus, the number of chromosomes in the gametes formed will be less or more than the normal haploid number of chromosomes, depending on which side the chromosomes go to. Alright? Either less or more than the normal haploid number of chromosomes. Right, the next question in this paper is on chapter 15 of the Form 4 syllabus, biology syllabus. So it's about sexual reproduction, development, and growth. Now, this question was asked only once in the past 4 years, and it's question 8. Alright, so you can try this question also from the past year question book. It's on the fetal vein blood, the blood in the fetal vein and the blood in the maternal artery. So a comparison was made. And then something to do with the seminal vesicle function. So it's a HOTS question. So it's asked in an indirect way. Then there's IVF and artificial insemination. So this is related to a HOTS question, and this question is quite difficult, quite challenging, right? So it came out in SPM 2022, right? So it's only come out once, so it can be asked this year too. So try the question to know the standard and how to answer it in the real exam. All right, try this question so you learn some techniques of answering. So refer to the past year question book. So let's go through the questions very quickly. This is the last question really. Form 4, Chapter 10, all right, on transport in the body. Diagram 8.1 shows an organ of a part of blood circulatory system in humans. State the function of artery X on the surface of the heart here, all right? Function, so artery X is actually the coronary artery, right? And the function, they just want the function. It supplies oxygen or glucose to the tissues of the heart or to the cardiac muscles here, all right? Or it transports oxygenated blood to heart tissues or cardiac muscles, all right? So as you mentioned, the most important one is oxygen to the tissues of the heart or cardiac muscles. Next, now nanotechnology used a nanoparticle that resembles high density lipoprotein or HDL. This is also found in the textbook. All right, this nanotechnology is mentioned. So nanoparticle resembles high density lipoprotein. Based on the above statement, explain how the HDL created can treat the problem of cholesterol deposited in vessel X. Okay, so that means you have to mention directly what the HDL does to the problem of cholesterol deposited in vessel X. So this needs some extra reading actually, right? Now, the HDL is the good cholesterol. It reduces deposits of cholesterol or plaque in the wall of the X. Okay, how does it do it? It will bind to the deposit of cholesterol and then carry it to the liver instead. That's why it's called the good cholesterol. Okay, it will get rid of the deposit of cholesterol. So then the blockage in the lumen of the artery will decrease because the HDL will take away the deposit of cholesterol or the plaque. The plaque is the cholesterol plus some things that harden it. Okay, harden the cholesterol like calcium. So blockage in lumen decreases when the plaque is removed or the cholesterol is removed, and the lumen size returns back to normal. So it goes back to a bigger size. So the blood flows more smoothly, and blood pressure decreases back to normal. Okay. So actually, it answers the question very easily, very directly. The HDL just reduce the deposit of cholesterol. That's the way it acts, right? So that the blockage in the lumen decreases, right? Then the blood will flow more smoothly, so it will be better for the health of the person. So use comparison mode again. Compare to a case where there is no HDL to help the person. Now, if an individual suffering 90% deposition of cholesterol in artery X, for this person, suggest a method how it can overcome the problem other than method in A2. Okay, so read the question again. For an individual suffering 90% deposition of cholesterol, that's a lot, okay, in artery X, which is the coronary artery. Suggest one method on how it can overcome the problem other than the method in A2 where you use HDL. Okay, so explain. So refer back, this is the kind of situation that are faced by many people who have cholesterol problem, serious cholesterol problem, where the arteries are almost totally blocked. The coronary arteries are almost totally blocked. So they have no choice. They cannot use the HDL treatment anymore. Okay, they have to go for operation. So let's see what it is. So there are three suggestions that can be used here. Okay. Now the first one is to put a stent, which is a small metal mesh tube, inserted into the lumen of the blocked artery X, like this. Now this is just for information, in case it comes up. So let's say this is the plug, the yellow part here. So first of all, they put in a tube-like structure, and it has a balloon attached to it. So the balloon can be blown up, can be expanded. Okay. Then it will press the plug. The balloon will press the plug, and the stent will also open up. Okay, the balloon expands the stent, which is the wire mesh here, and the plug is compressed. by the balloon and the mesh itself. Okay, so after that, this will increase the size of the lumen from this stage to this stage to keep it open for more blood flow. Okay, like in the normal case. That is the use of a stent, which is a small metal mesh tube used to expand. It's expanded inside the artery and then pressed against the wall of the artery. Okay, to press the plug to the corners and then to open up the lumen. Secondly, coronary artery bypass surgery can be carried out. Okay, what is it? A healthy vein from the leg is cut and joined to the coronary artery and aorta. Now here you can see the healthy vein from the leg, this one, taken from the leg. So it is free of blockages, then it is joined to the aorta here and joined to the coronary artery here to bypass this blockage here. All right, so that from the aorta, the blood will flow directly to the lower parts here, and the lower parts of the heart will get the oxygen. All right, if the blood were to pass through here, it will be blocked at the plug here. Plug will block the blood flow in the coronary artery, but it's bypassed because of this vein. So that is called the coronary artery bypass surgery. So a healthy vein from the leg is cut and joined to the coronary artery and aorta. This creates a new pathway for blood to flow and bypass the blockage in the artery. Okay. And thirdly, this is easy to write. Heart transplant can be carried out where the diseased heart is replaced with a healthy heart from a donor who has died. Right. Now, explain the effect. Okay, let's read these last few questions. Diagram 8.2 shows part of the system Y in humans with the lymph nodes. So this must be the lymphatic system. Explain the effect of the vessel of system Y, which is lymphatic system, in an individual's hand. Explain the effect if the vessel of system Y in an individual's hand is clogged. Okay, as shown here. Let's say the vessel of system Y, which is a lymphatic vessel, okay, in the hand is clogged. What is the effect? That means the fluid called the lymph cannot flow, right? If the lymph cannot flow, what else cannot flow? The tissue fluid from the intercellular spaces cannot flow. Okay, so the hand will become swollen. Now just state the effect first and then explain from there. The hand becomes swollen or experience edema, swollen because of the accumulation of tissue fluid. So the reason is the tissue fluid cannot flow into the lymphatic vessels from the intercellular spaces because of the clogging in the hand. Okay, we don't know how it's clogged, but let's say there's some clogging. So less tissue fluid or lymph is returned back into the blood flow. That means the tissue fluid actually has to flow from intercellular spaces into lymphatic vessels, but it cannot because of the blockage in the lymphatic vessels. So less fluid or lymph will be returned back to the blood circulatory system, and the tissue fluid that is not returned will accumulate in the intercellular spaces and cause the swelling. So there's limited bending or movement of arm. Now, it's only 2 marks, all right? So any 2 points will be accepted, okay? Okay, next question. Differentiate between blood circulatory system with system Y based on the following aspects. Now, system Y is actually the lymphatic system, okay? Because just now we saw the system Y with the lymph nodes, all right? So it's the lymphatic system. So the table is given to you, then you just fill it up. Now, what is the difference in terms of composition? One of the difference in terms of composition is blood circulatory system contains erythrocytes, all right? But lymphatic system does not contain erythrocytes or red blood cells, because all these substances, erythrocytes, platelets, and plasma proteins, are too big. All right, the substances or particles are too big to pass through the pores of the capillary walls. So they are not found in the tissue fluid, in the intercellular spaces, so they are not found in the lymphatic system. Okay, so any one of these substances, I'll just read through it. Blood circulatory system contains erythrocytes, platelets, and plasma proteins, whereas the lymphatic system does not contain erythrocytes, platelets, or plasma proteins. Next, importance of the blood circulatory system is to transport nutrients like glucose or oxygen to body cells, okay, and to eliminate or transport waste substances like carbon dioxide and urea away from body cells. All right. Now, the lymphatic system, it doesn't have all those functions. Its main function is that it produces lymphocytes for body defense, transport lipid droplets or lipid-soluble substances into bloodstream, returns excess tissue fluid into the blood flow to help maintain blood pressure and volume at normal range. Okay, you can just take any one. Maybe the first one is good enough because it's short. So for those who are interested, talking about nanotechnology, in case it comes up, so I just put some notes here from the textbook. Nanotechnology is used in the treatment of arteriosclerosis and plaque formation in arteries. There are particles that are designed to resemble the HDL, high-density lipoprotein cholesterol, the good cholesterol, to reduce plaque in the arteries. I like what we explained just now. So how does the HDL work? It removes the excess cholesterol from the artery and transports it to the liver. It will just pick up, like the trash man, you pick up the excess cholesterol from the blood and the artery walls and carry it to the liver so that the liver can remove it from the body. So in this way, the HDL is like the trash man, trash man that picks up the unwanted substances, the plaque, and then so that the arteries are not clogged, okay, and the blood flow is not blocked. So higher HDL level is good for an individual because it reduces the risk of heart disease, blocked arteries, heart disease, and stroke. Now, LDL is the culprit, is the bad guy. So HDL removes the cholesterol, but the high LDL, bad cholesterol, is the one that causes the plaque formation in the arteries, like this one here. Okay? Right, that's all for this lesson. I hope that you have learned something from it, and all the best for your exams. Bye for now. Now, tomorrow I'll probably post up one more video on winning strategies, number 3. Okay, don't miss that.